How are you? All are fine. I hope everyone fine at home. God bless you and your family. I welcome everyone back to my class. Today we are going to discuss about a new lesson, photosynthesis and respiration in plants. All organisms need food to meet their requirements of energy and to get the material they need for growth and maintenance. Most organisms also need oxygen to break down food to produce energy. As you already know, green plants, autotrophs. So, green plants make their own food by photosynthesis. The end product of photosynthesis are glucose and oxygen. Most other organisms dependent upon green plant for food. Example, heterotrophs, whether directly or indirectly. And most organisms include green plant need oxygen, which would get depleted if it were not continually renewed by photosynthesis. Carbon dioxide is essential for green plant to carry out photosynthesis. In this chapter, we will discuss photosynthesis and respiration in higher plants. Next, we move on photosynthesis. Photosynthesis. Pho means light in Greek and synthesize comes from a Greek word that means putting together. Is the process by which green plants make food using carbon dioxide and water in the presence of the sunlight. Plants have a power. Human can only dream of making food from sunlight. Sure, we can cook some savory dishes, but we use food already made, mostly by plants. This process of assembling carbohydrates fueled by the power of sunlight called photosynthesis. Keep in mind that sun emit all sort of different kind of energy, such as the heat and gamma rays. But photosynthesis specifically capture light energy. In addition, photosynthesis generated basic sugar molecules called glucose, which can be used to build more complex carbohydrates. Next, we are going to discuss about structure of chloroplast. A chloroplast is an organelle unique to plant cells that contain chlorophyll, which is what make plant green, and is responsible for enabling photosynthesis to occur so that plants can convert sunlight into chemical energy. So basically, without chloroplast, plants could not create energy, and without them, human would have no food and we would perish. So thank you, chloroplast. Structure of chloroplast. Chloroplast can be found in the cells of the mesophyll in plant leaves. There are usually 30 to 40 per mesophyll cells. The chloroplast has an inner and outer membrane with an empty intermediate space between. Inside the chloroplast are stack of thylakoid called grana as well and stroma, the dense fluid inside of the chloroplast. Plastids, the double membrane organelles found only in the plant cells. Chloroplasts are plastids that have chlorophyll and are vital for the manufacturing of food. Each chloroplast consists of two parts. The first one, grana. Second one, stroma. Grana, singular granum, which are piles of flanted sac-like bodies. Sac-like body means bag-like organ or body structures. And the stroma, a liquid matrix. The flanted sac-like body are called thylakoid. The thylakoid membrane contain protein complex that have the chlorophyll and its here that the photosynthetic reactions occurs. Take a deep breath in and then let out. Breathing to you, it's a very natural functions that you usually do without even thinking about it. When you breathe, you are taking in oxygen and releasing carbon dioxide. Taking in oxygen it's a very important because it allow your cells to do things like make energy from the food you eat. Plants breathe too, but they do it through tiny opening in leaves called stomata, singular stoma. Stomata open and close to allow the intake of carbon dioxide and the release of oxygen. It's a very important that they do this because this is the very oxygen that we ourselves need to breathe. Stomata are scattered. Scattered means spread among the cells of the surface layer, outer layer of the leaf. Some stomata occur in green stems as well. A stomata or stoma consists of two bean-shaped guard cells bordering the opening. Stomatal pore open when the guard cells draw water from the surrounding cells in the response to various stimuli, such as the intensity of light. Since the outer wall away from the pores, a guard cells is thinner than inner wall. The two cells bulge outward. They get swollen with water. This opens the stomatal pores, allowing for the exchange of gases, carbon dioxide, and oxygen. When the guard cells lose water, they shrink and close the opening. Stomata usually open during the day and close at night. Functions of stomata. The gas exchange that occurs when the stomata are open facilitates photosynthesis. Photosynthesis is the process by which plants convert sunlight into usable energy. During photosynthesis, carbon dioxide is taken in from the atmosphere through the stomata and oxygen released as a waste product. Both photosynthesis and the gas exchange that power it are essential to the plant's survival. Unfortunate side effect of the stomata opening it allow for water loss. Unlike you and me, plant do not need to sweat to cool off and prepare to keep their water inside. However, because the gas exchange of the photosynthesis is so vital, some water loss through the stomata is necessary. This process of the plant water loss is called transpiration. Although transpiration cannot be avoided, plant can minimize their water loss by controlling how wide their stomata are open, as well as what time of the day they are open. Opening stomata when the surrounding air is more humid means that less water will evaporate from the plant leaves, but opening them when temperature are warmer means more evaporation will occur. Likewise, if plant is already dehydrated, it may be close its stomata to prevent further water loss. Next, we move on factor affecting photosynthesis. Some of the factors that affect photosynthesis are light, chlorophyll, carbon dioxide, and water. In the following activity, you will investigate some of this factor. You will also see that oxygen is formed during photosynthesis and that starch is the end product of the process. Experiment to show that oxygen is produced during photosynthesis. The aim of the experiment to prove the oxygen is evolved in green plant during photosynthesis. Here use hydrilla plants. Hydrilla plants are water algae plants. This plant can bring out photosynthesis even they are surrounded by water. A large beaker, some hydrilla plants, one funnel, one test tube water, glowing splinter, hood and beehive arrangement. Next, we are going to discuss about procedure. The hydrilla plant should be kept in the beaker and covered with the funnel. Hydrola plant it's placed in such a way that its cutting ends remain toward the stem of the funnel. Water should be poured in the beaker in such a way that the stem of the funnel kept beneath the water level. A test tube completely filled with water should be inverted over the stem of the funnel. At this position, the experimental set should be placed in sunlight. Observation. After some time, it will be seen that bubbles are coming out from the hydrola plant and are accumulating in the test tube. After the accumulation of some gas, the test tube should be taken out carefully and a glowing splinter of wood be thrust into the test tube. It will be burst into flame. Now from the cut end of the plant, some bubbles are coming out constantly and they are collected at the top of the test tube by displacing the water. And the testing this gas, it is found that is oxygen. A bubble of gas it noticed in the experiment. Result, the liberated gas, oxygen, and it's evolved due to the photolysis of water under the procedure of the photosynthesis. The liberated gas comes in the intracellular space and eventually evolved out during the stomata. Conclusion. As the gas helped the glowing splinter to flame to the gas in oxygen because oxygen does not flame itself but help other to the flame. And second experiment. Demonstration of starch test in plants. Plants can synthesize their food. Animals including human cannot. Photosynthesis, the process by which plants prepare food using sunlight, carbon dioxide, and water. Sugar and oxygen are the product of photosynthesis. Sugar is stored as starch in the leaves. Experiment to demonstrate a starch test in plant. Presence of the starch in the leaves of the plants can be demonstrated by simple experiment. A few things are needed for this experiment. The plant, water, Bunsen burner, beaker, test tube with the test tube holder, thumb force, 90% of ethanol iodine solution. The procedure of the experiment is as follows. Take a potted plant. Keep the plant in a dark room for 2 to 3 days so that all the starch present in the leaf is consumed. Then take this plant and keep it sunlight for 4 to 6 hours. Pluck a leaf of this plant and dip the leaf in boiling water using forceps for 1 minute. Then boil the leaf in alcohol over a water bath till it becomes pale, hard and brittle. Again, dip the leaf into plain water to make it soft. Spread the leaf and add a few drops of iodine solution to the leaf. Following observation and conclusion can be made from experiment. The leaf will react with iodine and it turns bluish black. This shows that sugar is produced during photosynthesis and is stored as starch in the leaves. Hence, this experiment demonstrated the starch test in plants. Plants can prepare their own food by photosynthesis. Photosynthesis requires sunlight, carbon dioxide, and water. The end product of photosynthesis are sugar, oxygen, and water. The presence of the starch in the leaf it's indicated by the bluish black coloration of the leaf upon the reaction with iodine solution. Step of the experiment. Today I give 10 hard words. The first one, thylakoid, grana, stroma, stomata, God cell, epidermal, photosynthesis, hydrilla, chlorophyll, chloroplast. Once again, thylakoid, grana, stroma, stomata, God cell, epidermal, photosynthesis, hydrilla, chlorophyll, and chloroplast. Today biology assignment, learn, question answers. Thank you.